Hello everyone, my name is Sushant Kumar and I am a PhD scholar in IIT Bombay. Today I am going to present my work which shows that interleukin-9 promote malignant T cell survival by inhibiting oxidative stress and lactic acidosis in cutaneous T cell lymphoma. When priming of naive CD4 positive T cell occurs with antigen presenting cells, based on its microenvironment, it develops into diverse area of effector T cell subsets. These subsets are under control of specific transcription factors and secrete signature cytokines. Among these, Th9 cells are the new addition to the family of CD4 positive T cells, whose role is not very clear in many cancer types. However, there are several studies that show its anti-tumor role in solid tumors. Kruar et al. And Yong-Lu et al. Independently shows that increased production of IL-9 help in inhibition of melanoma tumor growth in murine model. In contrast, few studies have also shown its pro-tumor role in certain hematological malignancies. Cutaneous T-cell lymphoma represent a diverse group of non-Hodgkin lymphoma derived from mature malignant T cells that traffic to the human skin. On the basis of ACER registry data, the incidence of CTCL is 6.4 per million individual in the United States. According to WHO EORTC classification, primary CTCL include multiple variants. Among these, 66% of CTCL cases are mycosis fungoides, which is represented by erythematosus patches and plaques. The leukemic variant of MF, also known as Sézary syndrome, represents 3% of all CTCL cases. The second most common form of CTCL is primary cutaneous CD30 positive lymphoproliferative disorders, accounting for approximately 30% of CTCLs, which include primary cutaneous anaplastic large cell lymphoma and lymphomatoid papulosis. Subcutaneous panniculitis like T cell lymphoma is a rare form of CTCL, represent less than 1% of non-Hodgkin lymphoma cases, and localized primarily to the subcutaneous adipose tissue without involvement of lymph nodes. Earlier studies have reported abnormal cytokine profile and dysregulated T cell function during the onset and progression of certain type of lymphoma. However, the presence of IL-9 producing Th9 cells and their role in tumor cell metabolism and cell survival remain unexplored. Considering this, our first objective was to quantify the frequency of Th9 cell in CTCL patients. Secondly, we quantified the differential expression of IL-9 receptor on malignant T cells. And at last, we examined the role of IL-9 receptor interaction in metabolism and T cell survival in CTCL patients. This clinical study is registered in Clinical Trial Registry of India. We recruited 17 treatment-naive adult primary CTCL patients, provided they were not on any active immunosuppression at least 4 weeks prior to the sampling. Uh, Now coming to methodologies, after collection of human blood, the peripheral blood mononuclear cells from peripheral blood were isolated by five-fold density gradient centrifugation method. PBMCs were then stimulated in T cell media in a 24-well plate with PMA and inomycin in the presence of Golgi plug for 5 to 6 hours at 37 degrees centigrade. The PBMCs were first stained with fluorochrome conjugated surface antibodies, and then surface stained cells were fixed and permeabilized. Then stained with fluorochrome conjugated antibodies for intracellular marker like IL-9, IL-4, IL-17 and IFN gamma. Finally, acquisition and analysis by flow cytometry were performed for identification of Th9 cells. Here we are representing the gating strategy for the identification of Th9 cells. First, based on the granularity and size, we gated lymphocyte population, and then skin homing and systemic CD4 positive T cells are gated based on the presence and absence of CLA respectively. Further, Th9 cell population was identified based on the absence of IL-17, IL-4 and IFN gamma and the presence of IL-9 inside the T cells. Now coming to results and observations. The role of recently described Th9 cells have been poorly explored in CTCL. However, higher number of IL-9 positive cells has been shown to present in the dermis and dermoepidermal junction, which creates IL-9 enriched microenvironment in MF lesional skin. In the study, we quantified skin homing and systemic Th9 cells in early as well as advanced primary CTCL patient. As I have already shown the gating strategy to quantify the Th9 cells. So here what we have observed that in early as well as advanced stage CTCL patient, there was an increased frequency of skin homing Th9 cell as compared with healthy donors. However, higher number of systemic Th9 cells were found only in advanced stage CTCL patient as compared with healthy donors. Next question was the level of IL-9 in the blood of CTCL patient. So we examine whether patient-derived T cells secrete increased level of IL-9. For this, we have used ELISA, and we have collected cell-free culture supernatant of PBMC isolated from CTCL patient as well as healthy subjects upon TCR stimulation. Similar to flow cytometry data, we have found that IL-9 production was higher in CTCL patient as compared to healthy donors. So co-elevation of Th9 cell frequency and IL-9 production indicate that Th9 cells are the major source of IL-9 in CTCL patients. Next, to understand the functional relevance of presence of higher Th9 cells, we first examine the surface expression of IL-9 on malignant T cells. CD3 positive T cells were first sorted from CTCL patient as well as healthy individual. We observed the higher expression of IL-9 receptor on CTCL patient T cell and negligible expression of IL-9 receptor on healthy T cells. These data collectively suggest the co-expression of IL-9 and IL-9 receptor expression on malignant T cells. We further examine the functional relevance of co-expression of IL-9 and IL-9 receptor on malignant T cells. Live cell counting in the presence of IL-9 shown that IL-9 promote T cell survival of CTCL patient and had no impact on healthy T cells. Next, we examine the mechanism of IL-9 mediated increase in tumor cell survival. So we have performed apoptosis assay and found that IL-9 reduced apoptosis of the patient-derived T cell as quantified by annexin 5 and 7 ADD staining through flow cytometry. Since reduced toxic reactive oxygen species level are described to promote T cell survival, we examine whether IL-9 receptor interaction impact metabolic alteration, which could be beneficial for the survival of malignant T cell. For this purpose, sorted CD3 positive T cells from CTCL patients and healthy donors were cultured in presence and absence of IL-9, and then ROS level was quantified by DCF-DH staining using flow cytometry. Interestingly, we observed that IL-9 significantly decreased intracellular ROS level inside the T cell of CTCL patients. However, there was no significant reduction in ROS level in healthy T cell was found, thus indicating the role of IL-9 in maintaining a steady state ROS. Now, since lactic acid acidosis promotes ROS release and thus increase oxidative stress inside the cell, we quantified extracellular lactate in cell-free supernatant of CTCL patients T cell and healthy donor T cells. IL-9 decreased the extracellular lactate concentration in T cell culture of CTCL patients. However, it had no impact on lactate production in healthy donors. Collectively, these data suggest that IL-9 promotes cell survival while reducing the intracellular ROS level and lactic acidosis. Another interesting thing here is that ROS level have been shown to be elevated in various cell types when the extracellular environment is rich in lactate. The decrease in ROS thus could be due to this less acidic environment generated by IL-9. Now, coming to the conclusion of this overall work is that this study provides first evidence that in healthy individuals the Th9 cell number is less. When CLA on Th9 cells interact with E-selectin on the endothelial cell of inner wall of capillaries, it moves to the dermis where it secretes IL-9. But due to negligible expression of IL-9 receptor on normal T cells, there is no IL-9 IL-9R interaction, and thus there is no effect of IL-9. However, in CTCL patient, there is a presence of higher Th9 cells and elevated IL-9 in CTCL patient. Thus, here malignant T cell express higher IL-9 receptor. It demonstrates the autocrine positive feedback loop of Th9 axis in CTCL and delineate the role of IL-9 in reducing the oxidative stress and rate of apoptosis to promote the cell survival of the malignant T cells. Now, the crucial role of Th9 axis in CTCL pathogenesis indicates that strategies targeting Th9 cell might have a significant potential in developing robust CTCL therapy. At last, I would like to acknowledge my supervisor, Professor Rahul Pawar, for his valuable input, immense concern and suggestion, my alma mater IIT Bombay, our collaborator at Tata Memorial Hospital, Professor Hasmukh Jain, for providing support, my lab member, friend, family, Department of Bioscience and Bioengineering IIT Bombay, and all funding agencies including ICMR, DBT and WRCP. Thank you.